and meiosis, right? Mitosis, you have three functions, right? The first one is growth. Second one is repair and replace. Repair means that the cells are damaged, so you need mitosis to repair the damaged cells. Replace means that the cells are dead, so you need to replace those that are already dead. So these three functions, very important: growth, repair, replace, GRR. For meiosis, it's really just one function, is to make gametes, GAMETE. So gamete is just a fancy name for sex cells. Okay. Sometimes you're also going to see what we call a diploid, right? So diploid means that the chromosomes before and after the mitosis is going to be the same. You always have a pair of chromosomes in it. Meiosis, you're going to produce haploid. So basically only one copy of the chromosomes, right? Either the father version or the mother versions, or either one of them. So instead of two pairs, instead of a pair, you only have one chromosomes, one set of chromosomes. So for mitosis, you always produce identical cells. So identical cells with the same number of chromosomes. For meiosis, you're always going to create non-identical cells. Non-identical cells. And there are really just two types of sex cells, at the very least in eukaryotes: sperm cells or egg cells. And notice that in this picture, it also tells you some of the differences between mitosis and meiosis, right? For mitosis, you see that the nucleus is only get divided once, from one nucleus to two nucleus. The cytoplasm also divides once. So one cytoplasm becomes two cytoplasm. So the split of cytoplasm is what we call cytokinesis. Cytokinesis. Okay, make sure you notice the difference over here. For meiosis, the cells is going to split two times, right? First time is here, and then second time is here. Cytokinesis also happened twice. First time is here, second time here. How many daughter cells is it going to produce? For mitosis, one cell is going to produce two cells, two daughter cells. In meiosis, one mother cell is going to produce four daughter cells. So far, so good. Okay, next. The process of mitosis and meiosis interface to metaphase to cytokinesis. If you have a starting cells, right? Meiosis, both of them are going to have interphase. And during interphase, three things is going to happen. First, DNA is going to get duplicated. So you double the amount of DNA. The cell is going to grow bigger and bigger. And the cell also carries out its normal function. So these three things happens during interphase. And both mitosis and meiosis have interphase before mitosis and meiosis. Meiosis has number in front of it. Not in front, after. After all those phases, if you see the Roman numeral 1 or 2, you know that it is meiosis. If you see that there's no number after the phases, then you know that it is mitosis. For meiosis, can I zoom in? Oh, I actually can. Okay. So for meiosis, you see that the homologous chromosomes are going to line up in pairs. They don't really line up first. They pair out first, right? They pair out and they do crossing over. So they line up and do crossing over. Okay. How do I go back? Okay. So you notice that they pair up first, right? In meiosis, prophase 1, they pair up. So homologous chromosomes pair up. And they do crossing over. Crossing over is a very important thing because without crossing over, you don't have genetic diversity. Metaphase, the pair of homologous chromosomes line out in the middle. So they line out in the middle. And at anaphase, the chromosomes are getting pulled to the opposite side. And then in telophase, the chromosomes has reached to the opposite side. Has reached to the opposite sides, and you see that this pinching process is happening. And then after that, you're going to have cytokinesis. So cytokinesis, you're going to split into two cells. Then you have prophase 2, metaphase 2, anaphase 2, and telophase 2. The mitosis, prophase, and meiosis 2 are very similar, right? Prophase, you have the chromosomes that get condensed and thickens. Metaphase, they line out, each chromosome is lining out in the middle as a single line. Anaphase, sister chromatid is getting split to the opposite side. Right, sister chromatid is going to go to the opposite side. And then telophase, they have reached the opposite side. You see that the nuclear membrane is reforming, and then this pinching process happens again, and eventually cytokinesis. You have two cells now. Be able to recognize the picture. If you see that the homologous chromosome is the one that's in the cells, right? The homologous chromosome is still in the cells, and then they line up in pairs, you know that it's meiosis 1. Don't choose those that are 2. Right, in your video watching, I give you a picture and ask you which metaphase is this, metaphase 1 or metaphase 2. A lot of people say it's metaphase 2. It should be metaphase 1. Because if you see that the homologous chromosomes are lined up in the middle as pairs, it's meiosis 1. The number is 1. Okay, so this is the same thing again. The similarities and differences between meiosis and mitosis. Mitosis always produce identical cells. They always produce identical cells. Meiosis, they produce unique cells, right? It's genetically different. They don't have the same genetic information as the mother cells. Mitosis results in diploid. Meiosis results in haploid. Because mitosis function is for growth, repair, and replace, it happens all the time throughout the whole body except sex cells. So mitosis is involved in asexual reproductions. Meiosis results in sexual reproduction. In biology, A means none. So non-sexual reproduction. Okay, this is a very common question. What is the name of this process? Crossing over, right? You have a homologous chromosomes, right? So this is a homologous chromosomes. How do I know they are homologous chromosomes? Because homologous chromosomes are chromosomes of same size, shape, and function. You get one from your dad and one from your mom, right? One from your dad, one from your mom. You know that they started pairing up, right? The homologous chromosomes start pairing up. And this part is the part where they do the crossing over. How do you know that crossover? The green get exchanged to this side, and then the purple gets exchanged to that side. So this part, this touching point is what we call synapse. C-I-S. I might need to check the word again. I might get the spelling wrong. But this is where they cross over. Okay, next. Oh, before that, when does this happen? Mitosis or meiosis? Crossing over, yes.
It happens in meiosis.
Meiosis. Good. So it happens in meiosis. Meiosis 1 or meiosis 2? 1. Prophase 1, metaphase 1, anaphase 1, or telophase 1? Prophase 1, correct. So meiosis, prophase 1. Remember this. It is a very common question that will be asked. Next, similarities and differences between DNA and RNA, right? Everybody should be familiar with this already. Both of them have adenine, guanine, cytosines. But DNA has thymine, no uracil. RNA has uracil, no thymine. All these are bases, correct? The bases, the nitrogenous bases. DNA has the pentose sugar called deoxyribose. RNA has a sugar called ribose. DNA is double helix. So you can see that this helical structure over here. So double helical structure, right? Double helix. Okay, double helix. And then RNA is usually single-stranded. So they only have one strand, single strand. And their shapes are usually irregular. So DNA has 2 strands, RNA has 1 strand. DNA has double helix, RNA is irregular in shape. DNA is very big and stable. RNA is usually small and less stable. So far, so good. Okay. Next, you need to be able to identify the basic parts of a nucleotide. So a nucleotide is made out of a nitrogenous base, right? Usually we use a square to represent a nitrogenous base, a pentagon to represent a pentose sugar, and then a P to represent a phosphate group, or a circle, right? A circle to represent a phosphate group. Why is this always like moving around? Okay. Next, you need to know the bond. This is one nucleotide, correct? So this is one nucleotide. So the bond in between the nitrogenous bases, the pentose sugar, and the phosphate group, all these black solid lines are covalent bond. Okay, covalent bond. Okay, covalent bond. So how about this solid line then? Also covalent bond. So how about those dotted line then? We know that A pairs with T, C pairs with G. So when they're doing the pairing, right, there are actually those dotted line in between the bases. Those dotted line are hydrogen bond. Okay, those dotted line are hydrogen bond. Okay. Remember this pentagon is pentose sugar. Pentose sugar. Circle is phosphate group. And then the rectangular shape is nitrogenous base. Next, okay, functions and name all 3 RNA. mRNA stands for messenger RNA, rRNA stands for ribosomal RNA, tRNA stands for transfer RNA. All these are RNA, so they all have uracil instead of thymine. They're all single-stranded. There are different functions. You need to know the functions. Messenger's RNA's function is to contain the information or the message to code for protein. mRNA has a very unique thing on it. Every 3 bases is what we call a codon. If you did the extra credit, it says that how many bases are there in the codon, right? The answer is 3 bases. We always read the DNA bases, not DNA bases, the mRNA bases by 3, 3 by 3. We read it as a triplets. Each triplet is a codon. The codon pairs up to the anticodon, right? So the 3 bases on the tRNA is what we call the anticodon. So the anticodon is matched up with the codons, and each different types of codon has a complementary amino acids. So if you did your extra credit, it asks how many amino acids are there for one codon or for one anticodon. The answer is 1 amino acid. So what does tRNA do then? tRNA is going to carry or transfer, depends on which word you want to use, transfer amino acid to make protein. Where do they transfer it to then? They transfer it to the ribosomes. So the ribosomes is made of ribosomal RNA. So the functions of ribosomal RNA is to make ribosomes. So mRNA is going to be here. So you have the codons up here. And then we have the tRNA with the anticodon match up with the codons in the mRNA. The amino acids is going to start pairing up with the next amino acid. And eventually you're going to have a very long chain of amino acids. A bunch of amino acids bonded together is a polypeptide or protein. So far, so good. Okay. Okay, monomers of biomolecules. We learned this already. So the nucleotides monomer is, so the nucleic acids monomer is nucleotide. The carbohydrates monomer is monosaccharide. So basically your glucose. Lipid doesn't really have monomer, but it is made of fatty acids and glycerol for triglyceride. And then for protein, the monomer is amino acid. How protein is synthesized from DNA. So DNA, we're going to focus on the eukaryotes. So the eukaryote has nucleus, right? So this is the nucleus. This huge thing over here, this is the nucleus. So in the nucleus, you have DNA, right? The DNA. So first, you're going to use the DNA, which is your genetic information, the storage of your genetic information. So DNA's function is the storage of genetic information. So first, you have to base on your DNA in order to make mRNA. Reason, you need to make a copy of message. We call this message transcript. So it's like a piece of information. And then you're going to send this information out of the nucleus. So the process of making mRNA based on DNA is called transcription. Transcription. So transcription is going to use DNA to make mRNA. Then the mRNA is going to go out of the nucleus and meet with ribosomes. And then tRNA is going to come in and bring the amino acids. The process of tRNA bringing the amino acids and starts making protein based on this mRNA is translation. So remember, C first, then L. Transcription first, then translation. You need a script first before you can do the translating process. So far, so good. Okay. Next, you need to be able to transcribe a DNA code into mRNA, and then based on the mRNA, do translations by writing out the amino acid sequence. So during the test, I will give you a sequence of DNA, right? A sequence of DNA, and then you're gonna use this sequence of DNA, do transcription. Transcription means that you're gonna use this. A is gonna be paired to U, right? So U, C, A, because G pairs with C, T pairs with A pairs with U. So you're gonna write out the sequence to become the mRNA, and then during the test, you don't have to look for start codon, because I'm just gonna tell you this is the starting point, this is the ending point. You're just gonna start doing the first triplets, the first codon. So start reading it 3 by 3. Start reading the mRNA 3 by 3, and based on the mRNA's triplets, you convert it to amino acids. And in the test, the amino acids will be written as the first 3 letters in the chart. Yes. The chart?
Yeah.
Yes, the chart will be given in the test, and a physical copy will also be provided. So here's the chart, right? This chart, you don't have to panic. You're not going to memorize this, but it's very easy, right? Let's say UAC. What's UAC? So UAC. This is the first letter. This is the second letter. This is the third letter. So the amino acid that is coded for is tyrosine. So TYR. Let's say the DNA. Let's do the practice again. Let's say the DNA is AAA. What is the mRNA then? If the DNA is AAA, what is the mRNA? UUU, right? Because A pairs with T, but there's no T in the mRNA, so you have to change it to U. So mRNA is UUU. So you look for UUU then. So this is the U, and this is the second U, and then this is the third U. So phenylalanine, right? So the amino acid for this one is PHE. Pretty easy, right? Okay, next, you need to be able to identify and understand some of the key words meaning. So DNA, right, is a sequence of nucleotide with all those bases. If you have DNA packed with protein, it's gonna become strands of chromatin. But we don't actually use chromatin very often in our class. You just need to know that DNA plus protein in a condensed form is called chromosomes. Do you see that a long string, you start coiling them out and then pack them in a very closely together, it's going to become this giant structure called chromosomes. So remember, DNA is a segment of, a bunch of DNA coiled together with protein becomes chromosomes. So far so good? So this is the chromosomes. How do you count the number of chromosomes? You count the number of chromosomes by centromere, the center structure over here. So this is called a centromere. So here I have 1 chromosomes, 1, because I have 1 centromere only. How many chromatids? Chromatids is the arms. So here you see that I have 2 arm structure over here, so I have 2 chromatids. And you also need to understand, wait, let me see. Okay, never mind. So chromatids, 2 chromatids, 1 chromosomes. And chromosomes is DNA plus protein in a condensed form. Next, homologous chromosomes, right? When you have 2 copies of similar shape and size chromosomes. So this is considered as 1 chromosome, this is another 1 chromosome. How many chromatids are there in this picture? 2. 1 chromatids, 2 chromatids. They are called homologous chromosomes because 1 is from the dad, 1 is from the mom. And they are of similar shape, size, and functions. During interphase, right, this is gonna get duplicated. So it gets double. So this is really the clone of the previous one. See? So this is a clone. The purple is a clone of the previous purple one. We call this sister chromatids. So how many chromatids are there in this picture now? 4 chromatids. How many chromosomes? 2 chromosomes. Good? Okay. Next, you need to know what's a gene. So a gene is a segment or sequence of DNA that makes protein, that codes for a protein. So here we have a gene that codes for flower color and a gene that codes for pollen shape. And you can see that these two are exactly the same because these are clone of each other. So what functions you have here must have the same functions over here. Next, you need to be able to determine which name is homozygous dominant, which name is homozygous recessive. If you have a big T and little t together, this is going to be called heterozygous or hybrid. If you have a big T together, it's called homozygous dominant. Little t together, it's called homozygous recessive. Good. Okay. Be able to do the Punnett square. Know that if I have the same letters, right? B, big B and little b, I cannot write big B and little b in the same column. You have to separate them into one column that's big B, one column that's little b. Same thing, you have two traits over here. If I have big R, big Y, little y, you see that I don't write R and R together. R has to get Y in one cell over here. Okay? So this is another example here, right? And then once you did the Punnett square, you just look at the phenotype, what does it code for, and then you start counting the ratio. That's it. Okay.